Back there, and do we have signal? Okay, so we're rolling. All right, so what we're going to do today is we're going to continue that story from yesterday where we talk about phospholipid structure. We'd gotten to the point where we'd made the phospholipids. I'm going to repeat that today in the beginning because I made a mistake and tried to put two phosphates in, so I'll try to fix that. And then we're going to work through our way through phospholipid structure, and then we're going to imagine how these phospholipids are going to interact with water. I think in this section I got that far, but I didn't get quite that far in the second section. And we're going to talk about this in several separate steps. We're going to again imagine if we took those phospholipids and sprinkled them out on a planar surface of water. So if we had an absolutely flat surface of water, and we could magically put the phospholipids into a salt shaker and shake those phospholipids out onto the water. How would those phosphate head groups and hydrophobic tails interact with the water? Well, we'll see, of course, that the phosphate head groups will point toward the dipole moments in water. The tails will point away. So that's just a thought experiment. You can't really do that. But then you can imagine what's going to happen if we take those phospholipids with their hydrophobic tails and dunk them under the water. Well, the first level of organization you might think of is a micelle. The micelle is where the tails are pointing inward and the heads are pointing outward. The heads are in fact facing the water and the tails are pointing inward away from the water. The problem with that structure is it's not very stable. And I'll draw it and show you why. And the next level of organization is to make a bilayer. And so we'll make a bilayer of these phospholipids and we'll put them in a sphere, and that will be a liposome. These liposomes are actually quite important to us, particularly in this day and age. We're actually using effectively liposomes to make artificial membranes in which we encapsulate the RNA for the RNA vaccines. So we'll talk a little bit about a generic liposome and how we're going to take phospholipids, organize them in a bilayer, and form them into a sphere. And then we're going to simply imagine this sphere expanding and expanding and expanding, getting bigger and bigger, until finally the arc between each adjacent phospholipid is so small that it effectively is a planar bilayer. And this will be representative of what we see in cells. So then once we think about the planar bilayer, we can think about this as being a precursor to the biological membrane. Now, the biological membrane, sure enough, is a phospholipid bilayer, but that's not enough to be a proper biological membrane. To be a proper biological membrane and convey the idea of the lipid mosaic model of the biological membrane, those phospholipid bilayers have to be punctuated by transmembrane proteins. So we'll lay out this idea of making a phospholipid bilayer, and then we'll put proteins across them. One of the ideas I want to get across here is that the phospholipid itself, the phospholipid bilayer, is pretty much impenetrable by ions. Those fatty acid side chains are so hydrophobic that they don't simply allow charged particles like anions, cations to go through them easily. So then we have to postulate the idea of how are we going to get these ions across the membrane? How are we going to make a hydrophilic pathway for these ions? And that's largely going to be done by protein channels. But that brings us to the question of what's a protein? So we have to spend some time talking about proteins. And we'll simply talk in very straightforward terms, very simple terms, that proteins are nothing more than polymers of amino acids. We're going to see that there are 20 amino acids in biology. And they're different by just the kind of side groups they have on them, and we'll walk our way through that. And those 20 different amino acids in biology make all of the proteins we know in the world around us. And those proteins are formed by linking these amino acids by covalent bonds called peptide bonds. The chemistry of the amino acids allows us to make covalent bonds from one amino acid to the next, making a long chain, effectively a pop bead on a string kind of arrangement of making these amino acids linked out for hundreds or even thousands of amino acids. In order to understand this, we have to spend some time talking about the generic amino acid structure. What is it? What do all amino acids have in common that are permissive of generating these peptide bonds? And we'll talk about that. And then we'll talk a little bit about what makes one amino acid different from the next. And we'll see that there are 20 different R groups that we can put on these amino acids, and that will then confer the difference of one amino acid to the next type. We'll lay out the primary structure. This is going to be the amino acid sequence in the protein. We'll see that when we lay out the amino acid sequence in the protein, because bonds form, we get a kind of endedness to the molecule, so that every protein will have an amino terminus and a carboxy terminus. And that becomes useful to us because we're going to spend a lot of time talking about proteins in here, and we'll commonly refer to the amino terminus or the carboxy terminus of a protein. And if you read the literature, this is a common terminology that you need to be familiar with. And then we'll move on from the primary structure to something known as the secondary structure. Now, we probably won't get clear through this today, but hold on to the idea that the primary structure is just the sequence of amino acids, regardless of which ones we have, in the protein. The secondary structure is the next level of structure, and it's derived from hydrogen bonding. And we'll see that there's a positive charge on the amine, there's a negative charge on the double bonded oxygen, and those negative and positive charges can interact. That interaction, which is again hydrogen bonding, will give rise to two kinds of secondary structure, either the alpha helix or the beta pleated sheet. And we'll talk, hopefully we'll get to the alpha helix today and talk a little bit about that structure. But let me remind you where we were. We had generated a triglyceride. We had generated those ester bonds. Each one of the carbons on the glycerol was connected by an ester bond to the fatty acid side chains. And we'll put in our appropriate hydrogens here. And we said at the very end of the lecture that what we could do is we could leave these two fatty acid side chains intact. We're not going to mess with those. The number 3rd carbon we're going to fiddle with. So we'll basically say that on this molecule, this triglyceride, will have the same arrangement. Put this on out. And then on this carbon, we change it a little bit. We still have the 2 hydrogens, but now we use the alcohol, the oxygen of the alcohol, to link that oxygen to a phosphorus, to an oxygen, to something known as a head group. Now, when we look at this phosphorus, we see that there is a double bonded oxygen here and an oxygen with a negative charge. So we're already putting a fair amount of charge out here in this phosphate. And what's more is when we put the head group on, there are only 3, for phospholipids, there are only 3 of these we're likely to use. One of them is ethanolamine. Another is choline. And the third is serine. And what all of these have in common is they're charged. It turns out ethanolamine and choline are positively charged, serine is a slight negative charge, but nonetheless they're charged. So taken in aggregate, if we think about the head groups and the phosphate, this is a properly charged region. And where we ended was to say that we can have a kind of shorthand, where that shows the head group, and this shows the tails. And then we can say this is positively charged and emphasize the idea that this tail is. And then ask the question, how is this molecule going to interact with water? Well, in its simplest case, we can imagine taking a beaker of water and then having in our mind's eye and our thought experiment here, imagine here we have a kind of salt shaker. And in the salt shaker are phospholipids. And those phospholipids now, which look a little like this, when we shake them out of the salt shaker and they land on the water surface, if we could do this, this is not mechanically possible, but if you could do this, how would you imagine these phosphate head groups to be interacting with the dipole moments in water? Well, simply put, the head groups, because they're charged, are going to line up with the dipole moments in the water. And their tails are going to point up in the air. So this is what they call the lowest order thermodynamic state, favorable state in which these molecules can orient themselves. It's the lowest energy state. So that their charged head groups are pointing in the water, tails are pointing away. But that doesn't tell us very much about how these phospholipids are going to function as such an important component of biology. Because after all of biology occurs in a salt solution. So we have to begin thinking about how these phospholipids can live underwater. So let's imagine what happens if we take these phospholipids and dunk them under the water. How will they orient themselves? Well, the simplest thermodynamic state would be for these head groups to all point out. Because remember, this is water. All those moments that we talked about today. So the lowest thermodynamic state theoretically would be to have these head groups pointing to the water and the tails now pointing away. So this sort of structure where we have a single layer of phosphate head groups facing the water and the tails pointing inward, this is a name. It's called a micelle. You hardly ever see this in biology. It isn't stable. If you think about it for a minute, you'll recognize why it isn't a stable arrangement. You can imagine all these water molecules out here. Now and again, all you have to do is have one tiny water molecule sneak by the space between the phosphate head groups and get into these tails, so the whole thing blows up. It's just not favorable to have water around these tails. So this is not a biologically favorable structure. Now, we'll run into micelles again at the end of the course. When we talk about digestion, we'll see that the way we do lipid digestions, we do make micelles. But we don't make them this simply. We end up having a whole series of biological detergents called bile salts and bile acids, which help stabilize the structure. So this just isn't a very common structure in biology. It's just not very thermodynamically favorable. So how can we arrange those things in water, make it reasonably stable? Well, the concept that arises is something known as the bilayer. And the first way to describe the bilayer is in something called a liposome. These are pretty much artificial constructs. We make them all the time. They're quite useful in biology. But what they look like is they have a circle. I need to leave myself enough room. I hardly ever do. Those are the phosphate head groups. Out here, around the phosphate head groups, will be water. Now we know the heads are going to point toward the water and the tails are going to point away. Now if we had water in here, we'd have a micelle, so it would be unstable. So how are we going to orient these phospholipids in such a way that can make a stable structure? Well, the answer, of course, is a bilayer. And the way we're going to do that is we're going to have the tails pointing inward. So the tails, those hydrophobic tails, those fatty acids, are going to face one another. So the tails are going to face inward, and they're going to face one another. So this region, inside where the tails are, this is all hydrophobic. And these head groups, which are charged, some with serine, some with choline, some with ethanolamine, these guys are now going to face the water. Now, I think you can see that if we would generate this, you could safely put water in here. And if you can imagine this in 3 dimensions, rather than my simple 2-dimensional drawing, you'd have a sphere. The outer shell of the sphere could face the water on the outside. The inner leaflet of that sphere, made of phospholipids, now could face a droplet of water in the inside. Now, typically these liposomes vary in size, but typically they're less than about 1 micrometer. I want to think about, though, if we take this sphere and we make it bigger and bigger and bigger, so that we now have less and less arc between each of these phospholipids. Now, these phospholipids are tiny, by the way. These phospholipids are about 1 nanometer in diameter. So they're pretty small. Now, imagine if we would take this liposome and enlarge it to something the size of a cell, a biological cell. Let's use round numbers. Let's say the cell is, 10 micrometers in diameter. That's not an unusual size for a biological cell. Well, if it's 10 micrometers in diameter, you have to remember that there are 1,000 nanometers per micrometer. A nanometer is a billionth of a meter. I mean, we're getting into really small sizes. But you can imagine if we have a 10 micrometer diameter cell, and we want to know what its circumference is, we can simply say that the circumference is equal to pi times the diameter. Well, round numbers, right? 3.14, fine. But let's say this is 3. The diameter is 10, so that means it's going to be 30 micrometers, more or less, in circumference. Well, if each one of these phospholipids is a nanometer wide, it's going to take 30,000 phosphates, or phospholipids, to go around the cell at its equator. And I point this out simply because to say that there are going to be a lot of these going around the center of it. And if you can think about this in terms of the Earth, we know the Earth is round. Some people make the argument it's not, but the Earth is round. And if you go out here and stand on the lawn out here, you think it's flat. You think it's flat because to your eye, it seems to be flat. But in fact, if you imagine the diameter of the Earth is about 8,000 miles. Circumference at the equator is about 25,000 miles. So by equating these things, by thinking about these similarly, each mile has about the same curvature of Earth as one of these phospholipids. So it's not surprising that when you take this liposome and you make it bigger and bigger and bigger, that we end up with something known as a planar bilayer. So that for all intents and purposes, the bilayer appears to be flat. And in cells, what we have is effectively a planar phospholipid bilayer. So we'll simply change that liposome from above, extend it out, make it more or less flat, and imagine that there's going to be a tiny amount of arc. I'll let you do the calculations of how much arc there's going to be from one phospholipid to the next, but it's going to be pretty tiny. So when we think about the phospholipids and their organization, we can think about them in a cell as being a planar phospholipid bilayer. So here are head groups. You can imagine, of course, that there's water out here. Here are the tails. Put a bunch of tails in here, always two of them. And then, as we showed in the liposome, those hydrophobic tails are going to point toward one another. And now we have functionally made a phospholipid bilayer, and we can effectively make this into a cell. And we can say that on this side of the cell we have the outside, and on this side we have the inside. Now, I'll never ask you to know these numbers, but I think it's instructive to recognize that the width of this phospholipid bilayer is in the neighborhood of about 6 to 7 nanometers. It's pretty darn small. So we have this very thin phospholipid bilayer separating the outside world from the inside world. And yet, for biology to function, we need to be able to move ions from one side of the membrane to the other. And so in order to do that, if we have, for instance, sodium ions out here, or if we have chloride ions out here, these highly charged particles, the sodium or the chloride, either one of them, cannot easily pass through this hydrophobic region of the phospholipid bilayer. This is a pretty formidable barrier to these ions. And yet, for our nerves to function, for our muscles to function, for all of our cells to function, we need to be able to regularly and controllably move ions from one side of the membrane to the other. So the next character we have to introduce here is a transmembrane protein. And I'm going to show this transmembrane protein a little bit like a hot dog bun. And that transmembrane protein spans the phospholipid bilayer. And probably on Monday, we'll see that we can organize these proteins in such a way that they have charge. And because they're charged, this now provides a hydrophilic pathway across the membrane. This generic structure where we have a phospholipid bilayer, and periodically that phospholipid bilayer is traversed or punctuated by a transmembrane protein. Let me label this as a transmembrane protein. Trans meaning across, membrane, protein. So this is a protein that crosses the phospholipid bilayer. Biology, that we generate biological membranes by producing the phospholipid bilayer and then punctuating the phospholipid bilayer with the proteins. And that gives rise to a concept called the lipid mosaic model of the membrane. That's fundamentally how cells are built, how the envelope of a cell is constructed. It's constructed from this exquisitely thin layer of phospholipids punctuated by transmembrane proteins. Well, it's fine to say it's a transmembrane protein. It's fine to talk about them as proteins, but to understand how these things are going to function as transmembrane proteins, how we're going to make them into hydrophilic pathways, we have to spend some time. How is it made? So any questions up to here? Yes?
So my cell, just to be clear, it's considered a micelle when there's H2O that's penetrated through the, I should say, phospholipid membrane.
So what's a micelle? Okay, a micelle is a situation that I've erased. No, not here. A micelle is a situation where the tails are pointing inward, but now they're all interacting one with the other.
Okay.
And the heads are pointing outward.
So it's not a bilayer.
It's not a bilayer, it's a single layer. I have some figures here. I'll show you this. We've done that. Let me show you this one because I screwed it up yesterday. This is actually an image that was drawn by one of the TAs a couple of years ago. And it's trying to convey the idea of the phospholipid bilayer. Okay, fine. And the phospholipid bilayer is composed of these two layers of phospholipids. And then what Kyle did is he enlarged this to show the different kinds of phospholipids. And you'll notice that each of these three phospholipids, whether we're talking about ethanolamine, serine, or choline, each of these has these fatty acid side chains linked to a glycerol through this ester bond. So it's true for all of them. Where we begin to get into difference is here in the head groups. But in each case, we're going to modify that alcohol in such a way that we can now link it as an oxygen to a phosphorus to an oxygen. Then that oxygen is going to be linked here to an acetate group with an alcohol, with an amine on it. This is ethanolamine. In this case, it's going to be linked to the amino acid serine. And if you sit down and study this for a little bit, you'll see that these are the components of the amino acid serine. And over here, we have a similar arrangement, an oxygen, carbon, carbon. Now, rather than simply having an amine out here, we have this nitrogen with three methyls. And that gives this a sizable positive charge. So these head groups are now charged, and they are now capable of interacting with water. And that's the basic structure of the phospholipid. This is a pretty, what I think is a pretty picture of what I tried to put on the board in 2 dimensions. So put your phone away, I'd appreciate it. And what we see here is that we have a micelle. In 3 dimensions, the tails are pointing inward. You can see all we'd have to do is have one water molecule sneak by, and that blows up. It's just not stable. But on the other hand, if we make a bilayer, as we've shown here, where each of these white circles is a phosphate head group, the tails are pointing inward, we can arrange this in such a way that the tails are pointing and facing one another. That's stable. What's more is we can put water all the way around that, and that's stable. And you can look at this and see, well, this well could contain water, and that would be stable. And then we can take this phospholipid bilayer and expand it out to being planar. Just make it bigger and bigger and bigger so that we have a very small arc from one phospholipid to the next. And that way we effectively have a flat planar bilayer, and that's what we really basically see in cells.
Thank you.
Sure. Well, let's now start down the road. Are there any more questions before we start down the road to proteins? Yeah. Liposomes are generally thought to be artificial.
Okay.
They're generally thought to be something we construct. Anytime we have a liposome in biology, it's going to have some proteins across it. But when we make the liposomes, we can make them chemically, basically. And if you go into the literature right now, one of the really interesting studies that's been done is trying to figure out how we're going to make the proper kind of liposome to encapsulate the mRNA viruses, or the mRNA vaccines. And what you need is you need to have a phospholipid that has just the right kind of charge on it, so that when it encounters a cell, it will fuse with the cell and go into the cell. So there's a lot of experimentation done with the right kinds of phospholipids. And one of the bottlenecks in making them, apparently, is the amount of charge and the kinds of phospholipids they use. And this was an experimental technique that was done for years when people were trying to introduce different genes on different viruses into cells. So they developed those phospholipid bilayers to do just that. And they experimented for years trying to get those phospholipid bilayers just right so they'd fuse to a cell and deliver the cargo into the inside of the cell. That then was modified in mass production with these modern RNA vaccines. But they basically surround the RNA vaccine, the RNA that's meant to make the spike protein and the coronavirus, by those phospholipid bilayers. It's a liposome.
Okay.
Yes?
Just for curiosity's sake, does it then, like, would it be considered then a vesicle at that point? Or?
Oh, when does it become a vesicle? If you make it, it's a liposome. If the cell makes it, it's a vesicle. And again, if we talk about vesicles, and we're going to talk a lot about vesicles in here, vesicles have transmembrane proteins. As far as I know, there are no cases in biology where a vesicle is formed in proteins. Anything else? Yes?
Just to confirm, is the micelle considered hydrophobic because it would blow up if H2O was let in?
So the question here, is a micelle hydrophobic? I would say no. I would say that its components, now let me be careful here, its components are amphipathic. We're okay with that, because the head group and tails. But by the way I've defined hydrophobic, fair enough. It's not going to be stable in water, so it's going to be hydrophobic. But each of its, so the micelle itself, you might consider to be hydrophobic. Don't like the terminology, but I can see where you come to that. But the molecules that are composing, the composition of the micelle is made from amphipathic molecules. But the characteristics of water make the micelle itself unstable. Yes?
Clarify, basically it's easier for the phospholipids to create a bilayer than to create the micelle since it's unstable. So first, if you were to put it in water, it would probably make the bilayer first, right?
It's an interesting question. How do you make these liposomes? And I don't know how they do it. I don't know if it's just the thermodynamics, they mix these things together and they automatically go to a liposome, or whether there's something they do in their construction that forces that liposome structure. How do they keep the phospholipids just from being scattered throughout the water column? I don't know that. Someone knows that. Someone worked all this out. But I don't know how they do it. It may be patented someplace. Anything else? Proteins. Let's start with the idea of what are proteins, a simple definition of proteins. Now, when we talk about a protein, what we're talking about is a polymer of amino acids. Now, that raises the question, what's a polymer? A polymer is a molecule that's composed of hundreds or thousands of smaller subunits. And so in the study here that we're going to talk about, the cell itself is the amino acid. And the amino acid now is not living alone. The amino acid can be linked to neighbors by something known as a peptide bond. So we can define here a protein. A protein is a polymer of amino acids. One to another. By peptide bonds. Let me point out, it's important here, that these peptide bonds are covalent bonds. Let's start the destruction. Let's start the discussion with the structure of a generic amino acid. Now, a generic amino acid has the following characteristics. It has nitrogen and two hydrogens. that nitrogen is linked to a carbon. That carbon is linked to another carbon with a double bonded oxygen and an OH. You'll recognize this from the discussion of fatty acids. This is a carboxylic acid. This NH2 is an amine. Taken together, that's the name. This is an amino acid. Now, it turns out that in biology, here on our planet at least, there are 20 amino acids. All 20 amino acids have in common this amine group and the carboxylic acid. What distinguishes one amino acid from the next is what's covalently attached to this carbon. And I'm going to say that this is an R group. By saying it's an R group, it could be any number of things. In some cases, it's a single hydrogen, and that makes glycine. Some cases, it's a methyl, and I think that makes valine. Some cases, it's a ring group, and that's phenylalanine. Sometimes you'll attach a hydroxyl to the phenylalanine, and that's tyrosine. You can make all 20 different amino acids, and if you're in a biochemistry course, you'll memorize the structure of all of them. I won't ask you to do that. But I do want you to know that there's this generic structure of an amine group, a carboxylic acid group, and a carbon linked to an R group. The beauty of this chemistry, and we're going to talk about this in some detail when we talk about cell biology, is that we can now link one amino acid to the next. And I'll show some examples here. So the next amino acid here, and this is done in cells by ribosomes, the next amino acid, and I'll put it in blue, is again a nitrogen, a hydrogen, a hydrogen, a carbon with the R group, a carbon with a double bonded oxygen, and an OH. There will be something known as a dehydration synthesis, very similar to what we talked about yesterday in making triglycerides. And we can take the hydroxyl from the carboxylic acid, and we can take the hydrogen from the amine, and we can combine those molecules together to again make the OH and the H. Of course, that's H2O, that's water. And the water is derived from the hydrogen from the amine and the hydroxyl from the carboxylic acid. But in the process of generating this water, the ribosome in the cell, enzymatically, will make a new bond. It will link this carbonyl carbon directly to the nitrogen. So now this nitrogen, this amine nitrogen, is going to be sharing its electrons with the carbonyl carbon. When the nitrogen is sharing its electrons covalently with the carbonyl carbon, this generates a particular kind of bond called a peptide bond. The wonder of this system is that we can repeat this over and over again. So I'll show you some examples. Let's take another amino acid. There's the nitrogen, hydrogen, hydrogen, carbon, R group. Could be any of the 20 different R groups. Carbon, double bonded oxygen, OH. So again, there's the OH and the H. So we make H, OH. And the ribosome then has conveniently formed this peptide bond, the carbon-nitrogen peptide bond. And we can do it again. Let's try another one out here. Make it in green. Start with amino acid. Pull the hydroxyl off the carboxylic acid. Take the hydrogen off the amine. Make the HOH. And now again, form this nitrogen carbonyl carbon bond, which is a peptide bond. Well, let's do one more. Do I have an orange? I guess do an orange. Let's do one more, and then I want to point something out about this. Water H, OH, the formation of the peptide bond, and we can do this over and over again. And in fact, in biology, it's not uncommon to have a protein that's several hundred or even 1,000 or more amino acids long. So we can make this great long string of amino acids. Whenever we do this, though, it doesn't matter whether we have the 5 or 6 amino acids we have up here, or whether we have 500. There's always a conserved orientation because of the way we form these peptides. So we always have an end of the protein being the amino terminus, and the other end of the molecule out here with the carboxylic acid being the carboxy terminus. And this gives us a kind of language to talk about proteins. We can talk about proteins in terms of whether one amino acid or the other is closer to the amino terminus or closer to the carboxy terminus. And when you look at the literature, you'll commonly see the reference to this amino and carboxy termini. If you look at the literature on the COVID virus, they'll talk about the amino acids in the sequence, and they'll number them from the amino end to the carboxy end. So what we've done so far, though, is string together a series of amino acids, all linked by peptide bonds. That gives rise to something known as the primary protein structure. And the primary protein structure is nothing more than the amino acid sequence. Of the protein. And knowing the amino acid sequence of the protein tells us a lot about its structure. And with modern biological techniques, we can pretty quickly deduce what the amino acid sequence is in any done back out of the DNA or the RNA, but be that as it may, we will get the sequence of amino acids of the protein that will generate its primary protein structure. But that's just the first step along the road to trying to understand how this protein functions. Because what we're going to see is the protein function derives from its three-dimensional shape. I haven't given you much information about three-dimensional shape in here. There's almost no information there. But we'll begin to see that because of the characteristics to the R groups, we'll then be able to twist this molecule into a three-dimensional shape, and that three-dimensional shape will give it its function. But as we look at that, we can now move through levels of protein structure. We've described here the primary amino acid sequence, the amino acid sequence being the primary protein structure. The next level, I'll put this in the backboard, the next level of structure is said to be secondary protein structure. And the secondary protein structure is derived from hydrogen bonding. Within the protein. And that hydrogen bonding within the protein comes in two generalized types. The first type is said to be the alpha helix. And the second type is said to be the beta. Sometimes you see this as a beta sheet. More oftenly, you'll see it referred to as the beta pleated sheet. Now, before we move on to that, let me go back up to above to the primary amino acid sequence and remind you that each of these double bonded oxygens the double bonded oxygens will have negative charges on them. And these hydrogens, well, the amine itself, because the nitrogen is borrowing the electrons from the hydrogen, these hydrogens on the amine will have a positive charge. So the hydrogens on the amines have a positive charge, the oxygens have a negative charge. That's the basis of this hydrogen bonding. So let's think about this for a little bit. I'll try to start the story here of an alpha helix. So I'll do my best to draw an alpha helix. We abbreviate this as an alpha helix. And I'm going to show it as a kind of corkscrew. That's meant to be a helix. But that helix is composed of individual amino acids, and I'll put these in red. And we can number them. 1, 2, 3, 4, and 5. And I won't go, and this will be 6, 7, 8, 9, and 10. Well, why this protein? And we can put its amino terminus down here, and its carboxy terminus up here. Why this particular protein, why this particular series of amino acids takes on this structure, this spiral or helical structure, is because of the interaction between the negative charges on the oxygen of one amino acid and the amine on a neighboring amino acid. But it's not just next door. There's a formula, and the formula says that it's I plus 4. So if, for example, we think of I being the first amino acid down here, then it's going to hydrogen bond, make this number 1. It's going to hydrogen bond at 1 plus 4, so it's going to hydrogen bond to number 5. So 1, 2, 3, 4, 5. So number 1 will have its oxygen here bound to a nitrogen on this number 5. Number 2 will be bound to number 6. So the electrostatic interactions will again be the double bonded oxygen to this nitrogen up here. So the charge-charge interactions now, the lowest order for the charge-charge interactions is for the positively charged nitrogens and the negatively charged oxygens to align themselves that twist this molecule into the helix. Now this arrangement is such that it takes 3.6 amino acids to traverse the circle. Well, what do I mean by that? I think the easiest way to think about this is to think about a spiral staircase. We've all stood on a spiral staircase. We've all climbed a spiral staircase. You notice if you're on a spiral staircase, you start up the steps, and you're facing one wall, and you go up the steps, and as you go up the steps, you eventually come around facing to the same wall again. So you turn 360 degrees. To make that 360-degree turn on a spiral staircase typically takes 12 or 13 steps. But here what we're going to do is we're going to do it in 3.6 steps. So that means each time we take a step from one amino acid to the next, we start facing this wall, we step up to the next amino acid, we turn 90 degrees plus a little more, 110 degrees. The next one we turn 90 degrees plus a little more, and it takes us 3.6 amino acids to go 360 degrees. So that means we're walking up the spiral staircase, and each one of our steps is connected to, in the I plus 4 arrangement, by the hydrogen bonding. So think about that for a little while. We'll come back to this tomorrow to think a little bit about how the structure of this determines the structure of proteins.